Join me, Harriet Gould, for the Lab Matters podcast to hear fascinating stories every week from the inspiring people behind the science. In this episode of Lab Matters with me, Harriet Gould, I find out about the life and career of Professor Michlewski and how he would like to improve scientific publishing. Today we have with us Professor Gracjan Michlewski. Gracjan, you're professor at the International Institute of Molecular and Cell Biology in Warsaw, and you've done so much. You're so accomplished. I tried to think of, or tried to make a shorter way of introducing you, to try and incorporate everything that you've done rather than just that top line. But to be honest, there's just too much. So we'll have to work through it bit by bit. Thank you very much for being here with us today. Let's just start with what you qualified as. You got your PhD in habilitation from the Institute of Bioorganic Chemistry, the Polish Academy of Sciences in Poznan in Poland. And we went on from that, and we will go on from that, but perhaps let's just go back from that initially. And find out.
Thanks for having me, and I'm really flattered by what you just said. I always feel that I could achieve more, and so this is what drives me every day. But if we really have to go back, sort of, retrospectively to where it all started, I think when I was growing up, I always thought that I want to be a medical doctor, and this was something really sort of ingrained in my psyche, so to speak. And so I was fascinated by the idea of helping people, making a difference in their lives through medicine. So when I was 14, something really profound happened in my life, and my dad fell seriously ill, and then suddenly he passed away. And despite the best efforts of the medical team that was treating him. And at that point, I realized that while doctors play a crucial role in treating patients, there's still so many diseases that we don't really fully understand, and consequently, we cannot really treat them. And so this sense of helplessness drove me to really question the very foundation of our biomedical knowledge. And I think back then I really understood that I want to make a change. And it sparked my passion for scientific research, and I really became sort of determined to contribute to the field and to understand the diseases. And I think it's still with me to this day, kind of to push the boundaries of what we know and to explore biomedical science, ultimately towards improvement of people who suffer from incurable disorders.
What did your father have?
That was cancer, and indeed nowadays probably it would be even more better managed, but we still didn't really we won the war with cancer, so to speak, as a scientific community. But we are definitely closing in, so to speak.
It is, progress is encouraging, for sure. Yes. But that you're right, that is fairly profound, and it's almost like it didn't change your sense of purpose. It simply enhanced it, and you just wanted to do it even more. And even more effectively.
Probably, and what it also meant at a bigger scale, because when you're a doctor, you can help as many people, but it's still limited. Whereas if you do basic research and trying to understand the pathomechanism behind certain diseases, then obviously you can help many more. So the reward maybe is more difficult to achieve, but absolutely that was my thinking back then, and it still is.
It's very logical. And then you did your postdoc in the human genetics unit in Edinburgh. That must have been a culture shock.
well, not to that extent, but I think at that point where Poland entered European Union and when the UK was still part of this great team of nations, suddenly the doors opened for all of us. And since I was finishing my PhD and we were looking for options with my wife, who was also a scientist, the Edinburgh was there on the map, and essentially it was just pure luck that both of us got positions in the same place because of what you know as a two-body problem for scientists. So for us, it was a really easy decision, although anecdotally, I was visiting my brother who was already in the UK, but studying doing PhD in Newcastle. And then one day he took us to the trip, and it was the middle of May, to Edinburgh. And I just fell in love with the cities. It was so gorgeous, so sunny and so green and so warm. And so I thought, well, this must be the best place to live. So it was really an easy decision. Once we got our offers, then we were ready to move.
It must have been thrilling to get those offers in when you were so enchanted by the place.
Yeah.
So that was in 2005. And then you were there for a while. In 2011, you established your laboratory. Tell us about that and how that came about.
So obviously, you know, when I worked in the human genetics unit, we had an amazing time working in the sort of national laboratory led by Professor Javier Caceres, who's still my friend and my mentor until this day. And it was a time of essentially, you know, great discoveries in the RNA field. This is what I do. This is what I work on. But then I had urge to establish my own lab, because I thought that while being in someone else's lab, you know, you always be limited in what can be done, and you can explore ideas, but not to the same extent and not with the same kind of freedom that an individual lab head has. So at that point, I was looking for places to establish my lab, and Edinburgh wasn't the only place that I was looking at. I had offers from Berlin, from Bonn, from Manchester, but the Wellcome Trust Center for Cell Biology, which was part of the Edinburgh University, because the Human Genetics Unit back then wasn't part of the University of Edinburgh, you know, presented really with a great case for me to move and to fit this great organization. And I was quite lucky to get Medical Research Council Career Development Award, which really enabled me to set up my own lab and to work on my own topics.
Mm-hmm. And what were some of those topics that you worked on?
So in early days of my lab, we were focusing on the part of the genome which is non-coding. So, you know, for your listeners, I don't know how much do they know about the human genome.
They know everything, but assume I know nothing.
So we,
Tell me.
you know, we are built out of 3.2 billion base pairs of sequence of DNA. Yes, and then out of these 3.2 billion base pairs, we are coding maybe 20,000 protein coding genes, which actually constitute our body. But then there's hundreds and thousands, maybe even millions of the non-coding RNA, which is still coded by this genome. But they are the ones that actually, first of all, transmit the genetic information from DNA to protein. Those are messenger RNAs, but there are many others which control this process. And I was looking at certain RNAs called microRNAs. They are tiny RNAs, 20 nucleotide long RNAs, which actually control this process, and they're really critical in that they shape the determination of, you know, organs development and differentiation of cells, but also when they are misexpressed, they can cause diseases. And this is what was driving my research back then, to understand the biology and the regulation behind these small RNAs.
These RNAs differ from DNA by being single strand. Is that right?
Yes, that's true. So the DNA is the carrier of the genetic information, and this is compacted in this double strands with the complementary sequence. So the A will always base pair with T and the G with C. And then we have this sequence of, as I said, 3.2 billion of these blocks, so to speak. And then the RNA is just one strand made out of A, G, C, or U instead of T.
Mm-hmm.
And this is really just a carbon copy of the DNA. But this message is then carried to a protein, which is made of amino acid, and there are machineries coincidentally made out of RNA, which are decoding this information into proteins.
Mm-hmm.
And so this is really what we are doing, and trying to understand this process and how it's regulated in, you know, development of cells and differentiation of those cells, and also in different types of disorders.
So it's the RNA that's the really volatile part of the human structure.
I would say it's primordial. So there are hypotheses that the life began with RNA, because RNA can not only serve as the carrier of the information, as we see now with, let's say, RNA viruses, including SARS-CoV-2 or influenza or Ebola. Their genetic carrier is RNA, but RNA can also provide a metabolic and enzymatic function. So that's why, you know, people thought that maybe this is what sparked life on Earth, because it can do so much more than the DNA.
I wonder if they're right. So what would you say in your lab during that time was your greatest day?
You know, the life of a scientist, especially an active scientist that still does experiments, because by now I'm mostly writing papers, reviewing grants, you know, doing all the administration. But, you know, it's this discovery that you get a result, and you can, you know, at that point that you have a discovery. And this is something that I can really pinpoint to the day and to the place. And I know I was in the dark room developing something, and you know, looking at this X-ray film of whatever I just developed, I knew that I had a discovery in my hand, and that this is really significant, if that makes sense. So I had few of those, definitely. But what is also important to note is that scientists mostly work in the sort of in the shadow of the spectrum. So this excitement comes maybe once a year, you know, or twice a year, and most of the time we are troubleshooting. We are you know, trying to understand the experiments, why they don't work, what to improve, and how to make it better. And these discoveries and this, you know, sort of spark of realization that something is really, really important, they're the rarity of our life. But I think this is why it's so I don't know, addictive. That, you know, you want to have it, so you're prepared to spend another year, 2, 3, you know, just for this moment of, I don't want to say eureka, because maybe it's overstatement, but of, you know, of excitement, you know, of realization, you know, that you see something new for the first time that no one else has seen it before.
Yeah, I can imagine. Maybe not eureka, as you say, but certainly significant.
Yeah.
And meaningful, not just for you. We all do bits of work and we're proud of them, but to be able to be involved in things that are going to be so meaningful on a broader scale must be incredibly satisfying.
Yeah.
So 10 years or so there, was it roughly? And
Yes.
then and since 2001, you've been leading the Dioscuri, is that how you say it? Dioscuri,
2021.
2021.
Yes.
Dioscuri Center for RNA, the protein
Mhm.
interactions in human health and disease
Yes.
in Warsaw, in Poland.
Yes.
And that, I suppose, gosh, so you went there slightly during the pandemic.
It wasn't slightly, it was just in the middle of pandemic, but I obviously signed the contract and everything before, so we had everything aligned. And then, yeah, and then the pandemic happened, and then, we were kind of already too late, but not only pandemic, but Brexit, you know. So
Mm,
this is another complication that really put the spanner in the wheels, and I'm still amazed how we all kind of lived through all of it. But for instance, I can tell you a story that we were trying to move our goods, and because of the problem with Brexit and COVID, and all our stuff was stuck in Calais for 3 months. So essentially, me and my family, we were just living in the rented accommodation with nothing with us almost. So yeah, looking back, it was tough. But as I say, the offer that I got actually from National Science Center was again too hard, too good to refuse. So it's a money which is basically put together by the German government from Max Planck Institute as well as Polish National Science Center, and it's amazingly competitive and amazingly generous. So as I said, I committed and we had to move, but the whole process was was a big headache.
mm. And to win funding like that, to be awarded funding, it's like you say, it's a huge honor. It's not to be sniffed at. You can't really say no. But what goes into applying for that type of funding? What went
First,
into it for you?
first of all, you have to have a good idea. Also, you have to already have had successes in the publication sphere, so to speak. So that's also very important. And then finally it's, I would say, a bit of a lottery, because if you write something, you have some preliminary data, maybe it has to be judged by the panel members. And in the case of the Oscuri panel, it's really multi-disciplinary panel. So you have people from physics, chemistry, biology, medicine. So they're all of them basically judging. I think the head of the panel, Joachim Zauer, is a physicist who's a husband of Angela Merkel. Okay, so we are talking about
Nice
really
link.
level people judging you. But not only that, but then there are special reviewers, I think 6 or 7 reviewers all along the planet, so to speak, and they're judging you on the merit of your proposal. Okay, and then once you get to the second stage, usually you get to an interview, and then you have to come present your case, so that to pitch your case, what you want to do, and then obviously the panel deliberates with the reviewers' comments, and they make suggestions whether to fund something or not. That's how it usually happens. And the process, yes, it's very, very challenging, but again, if you don't try, you're not going to get. If people don't want to shoot on the target with the ball, then you're never going to score, I
Mm.
guess.
How long did it take?
So
The
the
process?
whole process usually lasts maybe half a year from when you submit to the stages of interview and then um, having a decision.
Hmm.
And coincidentally, I must say that I wrote this project, the Dioscuri project, to work on RNA viruses. And that was before the pandemics. So I got the money and now I'm studying RNA viruses, but it was just so timely and so um, unexpected that in a way I proposed something that is going to be not only relevant for basic science, but could be also relevant for you know, the whole human population, so to speak.
Hmm. You predicted the future.
Yes, almost.
Um, so you're still doing that. That's in progress, and um, and you're doing plenty of other things at the same time. Like you said, the desk work, you now judge for award funding. You do a lot of work away from the lab. How do you how do you fit it in for a start?
Uh, yeah, it's first of all, you have to have a good time management skills and a good calendar. And um, I'm not saying that I have everything perfect, but yeah, my calendar is full until sort of next half a year. Um, but no, I think this is sort of normal life. I don't want to sound exceptional. Like every scientist reaching this level, we accept it that we will be doing things pro bono, like reviewing papers, reviewing grants, um, contributing to teaching, even like okay, even if it's not part of your employment deal, I still do that. So that's something that is it comes naturally, I think, and I think it should.
Mm,
Um, but of course, there is a time when you say, okay, maybe I just did too much that I could chew. And for me, the moment was when I was on the board of editorial board of one of the Nature group journals, and I accepted with full heart, open hands, and I really committed to it. But the amount of work that was basically coming to my desk after 2.5 years, I realized maybe I should first focus on developing the lab here because that's more important. And then I can come back to the editorial sort of work
mm.
later on once I have a bit more time. So it's not like I can squeeze everything,
No.
we have to be careful.
Lab Matters is brought to you by the award-winning temperature control equipment from Polyscience, available worldwide. Polyscience, changing the world of temperature control forever. So, you said you're reviewing some grant applications. Are you allowed to talk about any of them? Have you seen anything that you think might be something that we'll all know about before long?
So I cannot really disclose anything because obviously this is it's not secretive as such, but I am bound by all the rules and regulations. I cannot tell one way or another. But what I more general, what I think medicine and biomedical science is going towards and changing is twofold. So first of all is personalization. The medicine will be personalized, and what I mean by that is that we won't be treating everyone as a sort of average of the society, but we will be actually sequencing their genomes, perhaps even sequencing their microbiome, and depending on the sequence, we'll be able to say whether there is some underlying genetic disease or perhaps some infectious disease that we can very, very rapidly detect. So that's one thing on the sort of detection and prevention side. But then there's another field which I'm really interested is the RNA technology, as we've seen, as we've witnessed with the vaccines against COVID. This was unprecedented in the way and the speed that this mRNA vaccines developed. And now what we will be using them is not only to vaccinate against flu or another strain of COVID and perhaps other sort of deadly diseases, infectious diseases, but we'll be using this mRNA technology to treat cancer, to treat those untreatable diseases, because it's the speed in which we can do the personalized detection of the genetic makeup of the cancer, and then the speed in which we could then generate the vaccine that would enable our innate and immune system to target this cancer. It's unprecedented, and this is the only technology that actually, for now, that will enable such rapid speed of sort of personalized medicine and personalized treatments. So that's, I think, what's really going to change in the next, hopefully not decades, maybe years.
I mean, I like to think that in a couple of years' time, we'll look back at generic medicine and almost laugh at it about how, oh, look how we treated everyone the same. They all got the same dose.
Absolutely.
Okay.
I think, well, there is still going to be generic medicine, I'm sure, because not all diseases are, life-threatening or whatnot. So it's more from economical point of view that governments won't be able to afford the personalized medicine for every single person. That's, I think, but I don't want to go there because I'm a scientist. I'm thinking in different terms than, let's say, government or big biopharma companies. But yes, I would hope that that's what's going to happen, and that we won't be able to. So we will be able to say really what's happening, and rather just to guess, because that's what most medical doctors do at the moment. Although my son is still in Edinburgh because he stayed over, and he's doing medicine. He is now on the 4th year of medicine.
You've inspired him, the next
And very
generation.
often I argue with him because he still thinks that medicine, as we know, is going to stay for a long time. And I say that you should be learning how to do genomic analysis and personalized treatment, because I think this could be the future.
Hopefully, if that happens and he hasn't learnt it, you'll be able to get him up to speed afterwards in your lab.
Maybe
You can spend some time in your presence. I'm just thinking there's probably 1,000 things that I should have asked you that I can't ask, that I haven't and won't, because there's not time. Before we finish, before I ask my last question, though, I just want you to talk about being a laureate of the Polish Returns program. Tell me what was involved in that and what it takes to be a laureate. I mean, other than what we already know about you.
Yeah. So it's essentially a granting agency that is attracting, I guess, Polish people that are sort of expats and achieved something really great outside, to come back and start putting in practice in Poland. And again, I was lucky to get this award. But crucially in my case, it was not about RNA vaccines or RNA viruses, but it was about Parkinson's disease. And this is interesting because it comes back to the first paper that I published with my lab in Edinburgh, where we were working on this, RNAs, and then serendipitously, a few years later, someone found that this RNA is key RNA involved in pathogenesis of Parkinson's disease. So So then, you know, we have to come prepared, and you have to be ready to venture into unknown. And I didn't know anything about Parkinson's disease, but I know how to analyze RNAs. And therefore now what we're doing with this so to speak, grant that allowed me to come back, but also establish some sort of line of research. That's important. I'm trying to leverage this information that we have found, the basic information, and to try to design small molecules or drugs that hopefully will be able to translate to clinic and help suffering patients, you know, from Parkinson's disease.
Mm-hmm. And that's another one of the big untreatables, isn't it? Parkinson's.
Yeah, precisely. And then, you know, we know so much more about the mechanisms, and this is really what I wanted to say, maybe from my own side, is we need to understand basic biology of a disease or method or tool or whatnot, because if we don't understand that, then it's just hand waving. And the minute we understand something, then we can try to design either molecules, different treatments, etc. And it takes time. You know, it takes time from this basic biology, basic biomedical biology discovery to translation. But there's no way around it. So if we don't fund basic biology and basic biomedicine, and we only focus on trying to force, I don't know, translational science or whatever it's called, probably the effects will be zero or very, very, you know, minor, because without the basic understanding of the process, it's like walking in the dark. And we basically yeah. So one message from this podcast, if I may, is that we really need to support basic biosciences, because those are really changing the field. And then, you know, as I was talking about mRNA vaccines, etc. This is decades of development, but without the development, probably many more people would have died.
Mm. So to move forward, we have to go back to basics.
Yes.
Mm. And how satisfying that work there that you're doing linked right back to some of your early work.
Yeah.
Before we end, well, you've already slightly answered this question. What are your hopes for the future of science in your area? So I'm going to change it slightly to if you could change one thing, about science, what would it be?
It's not. It's a great question, but I think it's a topic for
Reductive.
another podcast. But
Yeah.
there is a problem with publishing. Publishing in science is very structured, so that you have very top journals, Nature, Cell, Science, and when you get there, you can get all the credits, all the money in the world, and all the grants and allocates and everything. But these journals, they know that, and they exploit us, and they exploit society. So they charge huge amount of money for scientists to publish there. So we have to pay money. No, not we, but our grants, our institute, etc. Then they charge people to read them. Okay. And then they also ask me, whatever, to review for them for free. And of course, I'm going to do it because it's prestigious. And of course, this is part of our job. But just sitting on the side, I'm thinking that this is broken. That the system where the big journals or the, you know, the publishing corporations are earning margins of, I think, 50, 60%. which is unheard of in any other industry. And they're just basically abusing the system and abusing the researchers in submission, so that, you know, we all want to publish in these glamorous journals, but actually, I don't think it's necessary. But
So how
so
would
maybe
you,
I would change this to be more egalitarian in the way that it doesn't really matter where you publish, it matters what you publish. If that makes sense.
it it does make sense. I just wonder, because I mean, theoretically, anyone can publish anything anywhere these days. Everyone has access to a platform. The question would be how to get that quality of peer review to to verify that the publication, that paper is as meaningful as the authors say it is.
Yes, I agree, but this is really actually the problem that we have with the top quality journals, because at the end of the day, it's still 2 or 3 reviewers which are judging the paper or the manuscript. And so I think much better system is, you know, when you do post-publication review or peer review, and you allow the field to communicate. And actually it's happening. And actually that's why we're seeing so much retractions, because all these great papers now being analyzed by not 2 or 3 reviewers, but by hundreds of researchers. And they see the flaws and they see the data manipulations, etc., etc. So I think it's happening, but still, I think the attitude and the shift in the thinking about the quality of research, that's difficult. You know, if someone published in Cell Science, Nature, oh, he or she must be good, but that's not true. And yet we have this S program, yes, or something like that, which basically, you know, tells us not to look at the journal names or impact factors. But, you know, if you talk to anyone on the conference or anywhere else, they still do. So
Do you think open access will start to change those perceptions?
so open access is a great thing, but I think this is what I was trying to say, that the Nature is open access. You can have no open access, but you have to pay for it like £30,000 per publication or something like that. Really obscene amount of money that you as an author or an institute have to pay to these big corporations.
So you
So it is open access because people can access it, but at which cost?
so that's
So
the side of it you'd change.
yeah,
Interesting. So the ultimately market forces will determine what is a quality paper and what isn't, and they should all have the chance to be judged by those
yes,
markets.
I hope so.
Right. Well, we will monitor that and see how we get on. Gracjan, thank you so much for joining us today. It's been such a pleasure talking to you or listening to you. And good luck with everything that you continue to work on.
Thank you so much for having me, and yeah, it was my pleasure to speak to you, and I hope that I conveyed some messages. But yeah, thank you so much.